Okay, we'll go ahead and get started. So exam 2 is Thursday. We'll just do the regular round robin. Anything you have a question on, we'll take our time and go. If you need to skip. Okay, go take a call. Go. Okay, we'll start here.
I have a question about the XP experiment. Is it I don't know how to pronounce it, but the in the radioactivity buffer, there's in the bottom right where it's plus UV for the XP patient. Why is that one a dark spot? I thought that the NER pathway doesn't work at all in the XP patient, so the radioactive finding wouldn't get them.
So there's a deficiency,
Oh, not necessarily.
not necessarily, it has no NER pathway at all. So I guess the answer to the question, just to reiterate the question, on this whole wild type, this is the XP patient, you have ethidium bromide and the radio label. We have a minus UV condition, a plus UV condition, so minus plus V. We should have equal loading for ethidium bromide. We should have a little bit of baseline NER pathway being active, but DNA damage happens. Add UV, you basically cause a lot of damage. And a wild type has a really strong pathway, so you get a really strong signal because incorporating more radioactive nucleotides. In the mutant, they basically have less of this baseline, and then also less when you get UV damage because there is a deficiency in the NER pathway. Okay, so are we clear on that? Okay, we keep on going around the circle.
I have a question on one of the practice exams. There was the one where it says which of the following is a reason why specific elements producing restricting enzymes or multiple therapy methodology ones, and the answer was because CPNs can be temporarily helped by hydrogen bonds. I understand that, but I was not confused of one of the answers and why it was wrong.
Okay. But you can look the one you're confused on.
Because sticky ends created by any restriction enzyme can be ligated to sticky ends created by any other restriction enzymes. I thought like if we have the same restriction enzyme and then it cuts at like specific ends, you could like bind those two sticky ends together.
So what's the second half of that answer? Created by any
any
other,
okay.
yes. So just a reminder to very carefully read the answers. Because a lot of times on the exams, I'll go through the questions and I'll watch you guys in the back end. It's kind of creepy, like I'm a creeper. So I go to the back end and you'll toggle back and forth between answers, or you'll quickly select something and move on to the next. So you do this auto kind of like filling thing where, oh, I know this concept, so keep going. Even though you know a concept, take your time to carefully read the answer that you select or don't select. Because that one little tiny word made a huge difference.
Would this answer be correct if it said the same restriction enzyme?
Mhm.
Okay.
So remember, for restriction enzyme, the sticky ends, you want to make sure that you use the same restriction enzyme for the DNA that you're going to insert, and then for the cloning plasmid, so that the actual sequences can overlap, because it cuts out a very specific sequence. Yes. Okay, now we're your next person.
Oh. Could you, I know you just explained this one, but I was just a little confused on why the yeast is not for that last question.
Okay, so the interesting thing about yeast is that it's eukaryotic. And in the question from the very, very end of lecture, we essentially want to study bacterial infection of the library. And we know the mechanism includes bacterial protein getting into nucleus. So there are certain components of this question that can be asked and answered depending on what organism we want to use. So of course we can use bacteria because that's the thing that's affecting the fly brain. We can use fly because that's like the whole in vivo model where everything's happening. We use bacteria, so we get bacterial proteins. But the whole idea is that with the yeast is eukaryotic. The yeast has a nucleus, so we can study mechanisms of things getting into and out of nucleus using a very simple organ. So that's why yeast can be used.
Oh, question, number 4, there was a question about, setting up, yeast pads, but they used fungus instead. There was a question that said, instead of going, so I'm gonna play with calculating the values, it says, from X. you're going to play with medicine, which is the following sort of imply that compound oxygen mutagen. And then the correct answer is that your volumes are observed on the medicine play after exposure to compound X compared to the new compound X in. I was a little confused why that was the answer, just because the my interpretation of the image type, I thought like putting a mutagen in any code, given that we would have those because it is.
Okay, so let's go to the actual setup of this question. Read me the very first sentence, unless you can map it out here.
We would like to start with a test similar to the instance using the species accounters. Start with the providing oxytrol that is resistant to the antibiotic terramycin. We then test the
Pause, not that fast. And it's resistant to kanamycin.
Yeah.
Okay. Keep going.
We then test the potential for the subtle compound X and grow your plates on a plate lacking in the methionine.
Okay, so you have a methionine oxytroph, you have a plate lacking methionine. So it's also lacking methionine, I'm trying to understand the question. Plus compound X. Okay, anything else I'm missing from the question? So we're going to use a fungus. You said that it's a methionine oxytroph. And you can't make its own methionine. We said that's also kanamycin resistant. That's what we said, yes. What's the next sentence?
You test the potential compound X to grow your cells on a plate lacking methionine.
Okay. So we're going to grow on a plate lacking methionine. That shouldn't grow because it's an Ames test, right? And then if it's mutagenic, we should get some growth. And so let's check the answer, just make sure it's not mismarked. What's the correct answer on that question?
You called me so with after exposure to compound X.
Okay. Pause. What is that last sentence? What did we just say? What variable did we throw in? Read that last sentence again and tell me what variable is not written on the board somewhere in this experiment. So we said plus compound X, but then also kanamycin. And then we got fewer colonies. Okay, so our oxytroph, kanamycin resistance, methionine oxytroph. If we add kanamycin, what should happen? They'll still grow. If you cause some mutations, there's two possible things that can happen. You can revert this mutation or you can revert this mutation. Kanamycin resistance isn't normal. So by having kanamycin as the additive, and you get fewer colonies, we're basically losing resistance. And that's telling us that this compound is mutagenic.
So that's a mutation.
So remember, every single part of a question stem matters. And then remember when I talked about the thing about your brain kind of going into autopilot? By the time you got past Ames test oxytroph, you let yourself go into autopilot. And you went through the example as if it's the exact example from the slide. Without considering kanamycin, what would kanamycin do in the context of mutagen? Yes.
So this problem, what people are focusing on is the kanamycin, not the MET.
Yeah, so everything is plated on something lacking.
Lactic, so it's not
Yeah.
really variable for eliminating that.
Exactly.
Okay.
So remember, selection like I said, is gonna haunt your lives. So think about the genotype. Then when you have the genotype, make sure you know what this genotype is. Like what is the outcome? What will cause it to live or die? Yes.
I know that from your explanation, the mutagen caused the reverse mutation for kanamycin. Why are the other two answer choices incorrect? Or is the reverse mutation not possible for methionine in this case? Or why is it not possible?
Okay, so these are both grown on plates lacking methionine. So it's not that it's not possible for this to get reversed. It is possible, but you're not growing it in a condition that would differentiate. I mean, so to get, so like you have to reverse both. So the way it's growing, like, how do I make this clear? Both of our plates are lacking methionine. It can't make its own methionine. And then so you'll get some growth if there's a mutagen, but you have a second gene that you have to account for. So normally, if I were to draw this out again, I would probably give myself a 4x4 grid. Because I have two genes. Let's do this. This is compound X in the middle, compound X in the middle. And then let's say the bottom half is going to be if it's yes mutagenic. The top is if it's not mutagenic. We know that everything is going to have, they're going to be lacking methionine, lacking. Everything's gonna be lacking. Using these types of mutations, you can get a little bit of spontaneous mutagenesis, so reversion. So only the way you see like some stuff growing is if you have some of the reversion mutation occurring. This can be basal, or this can be like specific to whatever we put there. But say we add in this other variable, kanamycin resistance, and we decide, you know what, I'm gonna add kanamycin to half my plates. Without kanamycin, it's not gonna make a difference because we're not adding it anything to kill it. But if I add kanamycin and there is like this resistance, and say it wasn't mutagenic, it's stay the same. But then if I lose the resistance, I lose colonies. So that's causing more mutagenesis. So the fact that we have less colonies in the presence of kanamycin is telling us that compound X is more mutagenic. Okay. Two genes is track both genes. Other thing I want to warn you of, I try to make some of the examples a little bit more difficult than the exam, so you can think through the whole process. I won't give you a number for A's. Maybe I shouldn't. But you've been warned. Okay. Any other question on Ames test? Like you do need to know Ames test and how it works, how to read it, what tells you whether or not it's a mutagen or not.
I mean, I have one on Ames, so I have another question, but there's on the subject.
So you're on the subject, let's go.
For a positive and negative control, what was the difference of them again? A positive and negative control.
Okay, so not necessarily, what's the last? It's not necessarily positive and negative control, but you'll have for the negative something that doesn't have a mutagen. So no mutagen versus like if you're setting an experiment, I would also do a no mutagen. Most people replace the no mutagen part with dilutions. And then what you want to test, mutagen. If there's no mutagen, you don't expect to see the reverse mutation. A couple of them are spontaneous, you'll see them like randomly throughout the plate. If you test mutagen, this is when you expect to see, if it is a mutagen, you'll get growth. And in a circle, or some type of like clear zone. When you do dilutions, you know it's dependent on this mutagen because your clear zone gets smaller as you dilute it. And then if you wanted to make sure that you just know how to do an Ames test, it's always good to have something that you know is a mutagen to make sure that this reverse mutation is working properly. So this is how I would set up negative control, positive control, or you set the negative control, you test mutagen, and also have dilution.
So positive control is like kind of like what you know is going to happen. Negative control is kind of like without anything in it.
Yes, exactly. The negative control, like without intervention, without stuff. Positive control, what we expect to happen, and use something that you know will cause that.
So like one's like what we expect to happen with stuff, what we expect to happen without stuff.
Yes, yes. Yes,
So for like in no mutagen, you see no growth, that it's not mutagen, or you always see something.
and where?
And then,
And then no, so because there's spontaneous mutations, usually you always see a little bit.
okay, but to confirm that whereas in Eugene, involved, you see a pattern of like peripheral. Like,
Yes. So we'll always see a little bit of like spontaneous reversion mutation. But to confirm it's a mutagen, you see this growth in a pattern, also with the differential sizes of the clear zone based on their dilution or concentration used.
And then confirm that by the body control.
Yes, positive control ensures like your oxytrope is working, like I'm not just an idiot, like. I think use positive controls for. Yes,
So the more diluted your region was, it'd be more diluted, there's gonna be smaller.
what do we think?
Yes.
Yes, the more dilute the mutation is, the clear zone gets smaller and smaller because it's less intense. So what causes this clear zone in the first place? So much mutation that would cause cell death. Because inevitably, if you're gonna cause mutation in something important for life, cell not gonna join. Any more questions on the Ames test before we hit your main question? Okay, hopefully, yes,
So it's only for, so the mutagen, the clear zone, basically like still the cell that overwhelming with mutation.
exactly.
Okay, all right.
So high dose. And then y'all know how the fusion works, gets to a lower dose as you radiate out from the center of the plates. Okay, cool.
Yeah, so my main question was regarding, it was the mismatch repair question pathway, where it was that case where the C is the, where C is the final nucleotide added to the mismatch repair pathway to fix an error. Where will the ligase act and why? And we see that it's gonna be between like the C and A, it's gonna be on like that, so that I understand because of how it's extending, because DNA extends 5'3" prime. But what I don't understand is where it knows where to cut. So like that hemimethylated site, I was wondering if you could draw out like where it would be on that sequence like that, and like kind of how it would determine based off that. So I'm confused because in the stuff it says a methylation occurs and then snips the non-methylated site. But then how do we know where that is in reference to where this ligase is working or where this
Okay,
is?
so for this, we're just tracking backward and we're not going to spend a whole bunch of time on it because I'm not going to test you on this on the exam.
Okay,
But say, for example, what we had in the notes is that I told you what was the very last thing inserted. And remember we said 5', 3', I had it on in that direction. And ideally, if we're sequencing that way, it means that on this side, we figured out that there was a methylated site somewhere over here
on the left.
side, because we started sequencing in this direction. That's really the only thing that we can glean from knowing that this is the last thing inserted. Because of the 5'3" direction of synthesis.
So what's your gap C sequence? Is it also on the left?
Yeah, it's on the left, So that sequence is where it methylates, and that's what it identifies. So it's, yeah, it's some of the things I love about these mechanisms of cell molecular biology. We'll get to a point where we're like, we know what happens, but we're not 100% sure, like how. And this is one of those. So you have, this is one called all the meat proteins, right? You asked me about meat.
Was that?
That's one called meat proteins? Yeah. So they know that it can differentiate, or technically not know. They assume that it's differentiating based off of the lag and the methylation. But that's still technically a theory / hypothesis that has never clearly been shown to be the case.
So then just to clarify, like when this, so for prokaryotes and whatnot, like for mismatch repair, you methylate a strand that's targeted, and then it comes in and snips the unmethylated side.
That's the unmethylated side. So the whole idea is that there's just a lag in methylation because you're making a new strand.
Which we talked about.
Yeah, and then so because there's that lag, that's not going to identify which one's a daughter strand.
And do this, okay.
Like so, and the whole mechanism of eukaryotes, they're not even sure about that either. I can go through a couple lists of things of like, yeah, they don't really know.
I think I'm overthinking it, but yeah, I get it. I think
Okay, cool.
Okay, I had a question a week for homework. I think I'm just having trouble like picturing it, but it was about how homologous recombination could be used in the lab to replace a gene of interest with a selection marker. And then the question was, if a cell is capable of, is also capable of non-homologous end joining, and which of the following would you expect to be true if you're transforming a selection marker into a cell that can perform both homologous recombination and non-homologous end joining?
Okay, so I have a cell that can do two things. It can do homologous recombination, and it can do non-homologous end joining. So the initial question, what's the setup and scenario? Write out these points.
So-
First sentence.
a cell is also capable of non-homologous end joining, so it's capable of that. And then it's, which of the following would you expect to be true if you're transforming the selection marker into a cell that can perform both?
Okay, so we're putting a selection marker into a cell that can do both of these. So remember we talked about, transformation of a plasmid, and then depending on that plasmid has the ability to get incorporated into the DNA. That's kind of the topic that is going off of. Read to me correct answer, we'll go over why it's correct, and then wrong answer, we'll go over why it's wrong.
Okay, so there's 3 correct answers. So those were transformed cells would still be able to, could still grow on the selected media. The gene of interest could still be knocked out, and the selection marker could still be transformed, but may be randomly inserted into the genome, not necessarily replacing the gene of interest.
Okay, so the first one is basically the transformation. Like works fine, it can express the selection marker, so that's thinking that this plasmid got into the cell and then it can still replicate the plasmid. The next one is that because it can do homologous recombination, whatever the plasmid looks like, it has the potential to actually like knock out a gene of interest and incorporate into the actual like genetic material of that cell. That's why 2 is correct. Number 3 is that could get randomly inserted. If you're going to get randomly inserted, it's because you have the non-homologous end joining, you cut it any place and it kind of puts them back together. So it's framed up. And then, so now let's go with our incorrect answer.
And the incorrect answers were transformed cells would no longer grow in selected media, and the selection marker can no longer be transformed into the cell.
Okay, so why would, well, if this one is correct, if you can transform and still grow, why would it not be able to grow? So again, if you can express it, no problem, it's not be able to grow something media. Sounds, okay, and then the second one, is there anything that's happening here that would impact the ability for transformation to occur?
No.
No,
It's talking about a little bit with models of combination was, what was the purpose again? You were saying like why you want to knock out a gene specifically, was just to see what it does, or is there other reasons?
that's what most. So there's a lot of papers where we're like, you know what, we found this new gene, it's associated maybe with disease, let's see what it does. So let's see what it does, or see what it do, and just knock it out.
Is there any other specific things to know, or that's mainly just?
That's mainly it. You should just knock out a gene so you can see what function it has.
Thanks so much.
Yes.
So this question was phrased where it was just a non-homologous end joining. Would the gene of interest still be able to be recombined and then transformed into the cell? And with that transformed cell, would still be able to grow?
So transform cell will be able to grow, you'll be able to do gene of interest knockout, but if you don't have non-homologous end joining, you're not going to get random insertion.
What if like it was just non-homologous end joining, like we had no homologous component?
So then you'd actually just swap R, N, E, J, and this would turn to a yes, and this would turn to a no. You wouldn't be able to knock out the gene of interest.
But then in that case, like we just don't have no transformation.
The transformation will still happen because it's a plasmid.
It
It'll
will
stay in the
show
cell.
up like in the slow reaction because we're not having the. So it just wouldn't show up in the plasmid.
Well, the selection marker is being transformed in, so the selection marker is still part of this plasmid. It doesn't have to be incorporated into the DNA or
So it just doesn't replace the gene of interest.
interest. It should be
It
knocked out.
replaces like a random.
Yeah, or just, it just ends up anywhere. It could replace something, it could destroy something, it could just end up in a place where there's no coding. Random. Okay, so how are we on homologous combination, non-homologous? Or I can't write letters. Non-homologous end joining. Yes.
I was wrong here for homologous recombination. Does it just not occur at all during G1? Like, does it require the replication like to be done, or can it be like can the homologous chromosome be used as a template for homologous?
So it's very rare that it happens during like G1. It normally happens during S phase. Because so technically you can use other chromosomes. But you normally have an ample amount of accessible DNA there during the S phase when it's replicated with chromosomes.
So, oh sorry, before it's replicated, you have mostly and homologous end joins, and then once it's replicated, mostly homologous recombination.
Yes, everyone's clear on that. Replicating, homologous recombination. Not replicating, way more non-homologous end. Know that, wait. Okay, Yes.
Like, could you do that again?
Okay.
Sorry.
During replication, S phase, more homologous recombination. During resting phase, or like G0, the cells depend more on non-homologous end joining. Know that wink, wink, hint, hint, okay.
Okay, so HR is for S phase and then NHJ is like anything but S phase or just outside of the
Basically
subset?
outside of subset.
But even during G2, would you have mostly non-homologous things in?
Yeah, so I would think of like your HR associated with S phase. And then don't get too hung up on like G2, G0. Like, it doesn't matter, know the dichotomy. Okay. So we went around the middle table, we'll start going around the outside edge. Any questions from the outside circle? Or shall we skip and come back? Just let us know. Okay, so model organism. I'm gonna go over this one more time. The idea is that the question that we have is on bacteria, infection, and a fly brain. And then we believe the mechanism includes transports of bacterial protein into the nucleus. I'm coming across a question of what model organisms can I use. What I would like you to do is think of the organisms that are being studied or that can be used to study a particular function. One model organism does not necessarily have to answer the question completely. So for example, in my laboratory, we're interested in type 2 diabetes. We use mouse, we use human cells. We can't use a whole human, so we just use the cells. And then we use the mouse to replicate like growth and obesity to look at things like that. And then sometimes we'll use, let's say like a bacterial vector, because we want to express a protein that we want to look at. And the protein can be expressed through the bacteria. So it's two different organisms in order to model what's going on in humans. So here, we're like, okay, bacterial infection is a thing. So check, we can use bacteria as a model organism to answer a portion of this question. We're concerned about fly brains. So check, we can use flies. And then the third one that I think is getting a lot of people tripped up is the fact that we can also use yeast. But the reason why we can use yeast is because it is eukaryotic. Forget my spelling. I can't spell, I can't math, but it's fine. Eukaryotic. It has a nucleus. Because it has a nucleus, we can use it to understand basic mechanisms of transport into the nucleus. And that's why yeast works. So think about it. All the things we know about genetics and replication and the genome and sequencing. How much of it did we actually learn from experiments with virus and bacteria, Hershey Chase? It had nothing to do with humans, but yet we learned what our genetic material was because of a virus and a bacteria. We use bacteria to learn about replication of DNA. And so much of what polymerase 1 does translates over to human systems, even though they're more complicated. So this is what we mean by being able to identify a model organism that can address the question being asked.
So virus would also be a correct answer as well.
Oh, for this one?
Yeah.
No, not for this one.
Oh, no.
Yeah, as well, yeah. Yep, no, not for this one. Yes, question and question.
So basically, like any organism that's easily grown in lab and has a nucleus and material to work with it.
Yeah, for this, yeah, I want to say check right, it's nucleus, basically any eukaryotic system, probably works just fine. Okay. Oh. Well, yeah, I'm thinking of like, hey, okay. Next question from the outer rim. Or Pacific rim, y'all see that movie? Oh, question on this topic? Okay. Oh, yeah, we're going around this way, we said a couple people.
You never had it before.
Oh, okay, we'll go out of order.
It's okay. Oh, okay. I'm sorry.
Don't apologize.
I just had a quick question, like as far as like just clarity on sensor sequencing. Regarding CPS, because earlier saying like, since there's no oxygen there, like, can just send them stop.
Yes.
So like. I just wanted to see if we could go over like some scenarios that would help us understand. And what situations in where you're at.
Okay, so I think the biggest thing here is to just really focus on what's the role of the double deoxynucleotides in standard sequence. So for example, we're gonna have a template strand of DNA that we care about. And we don't know what the sequence is. We want to sequence it. And to sequence it, we're going to need some type of primer. If you don't know like the beginning or like say for vectors, a lot of times we can like make a primer to a part that we know. You don't know at all, people use a bunch of random primers to see what they amplify. It's kind of like the next generation sequencing. But you want to make a primer. This primer is going to be labeled, usually radio labeled. And then because we extend from 5' to 3', we're interested in being able to sequence this template strand by making the complement. So what we take advantage of is knowing that in the mechanism of DNA extension, you have to have that 3' OH to nucleophilically attack the phosphates and methylates. So we incorporate what we call the DD NTPs. The DD NTPs basically don't allow for nucleophilic attack because we do not have a 3' OH. No 3' OH. No nucleophilic attack.
Oh, do you do meta sequencing or not?
You do not.
Oh,
No DNA extension. Okay, this is what that means. When you do the reaction for Sanger sequencing, you mix a little bit of DDNTPs with a lot of it of DNTPs, the ones that work normally. And this is important. So I'm going to do one example, and we're going to start off with just adenines. So what happens because we have a lot of these and a little bit of these, as DNA is extended, sometimes it'll get somewhere and it'll incorporate a regular A, keep on going, incorporate an A, keep on going, incorporate an A, and then, oh, I accidentally hit one of these. Now I'm stuck. Another time it could do it, and then get stuck here. Another time, in all the same reaction, this time I get stuck on the second A. So you basically have a mixture of these different lengths of nucleotides because every now and again they're getting stuck in DNA extension. So now when we go, we run this whole mixture on a gel. Oh my God, okay. Run this whole mixture on a gel. Arrow. The one that's really, really small runs all the way to the bottom of the gel. And we have a signal because it's radio labeled. Something that's a little bit bigger, well, it'll appear like somewhere here. And the really long one appears towards the top. And then every place that we have a band or a signal is a place where this DD ATP got incorporated, giving us the actual nucleotide that was incorporated. So now what happens is that we do the same reaction for the other 3 nucleotides. And that's what gives us the total of 4 lanes for each one. Are we clear there? Yes.
Are the primers the ones that are labeled or the DDNTPs?
The primers are the ones that are labeled, but do you think it would work if the DD NTPs were labeled? Would that work? Yeah, it'll work. Yes.
For knowing the difference between like just like PCR and Sanger sequencing, it's basically the main difference you would say is its use of DD NTPs, and I'm looking at that as well as yeah, pretty much makes sense.
Screen. Screen's over there. Okay, I'm gonna erase this gel. I think this part makes sense. So we were asked about some of the differences between Sanger sequencing and PCR. So let's map some of these out. The Sanger sequencing, PCR. So first thing about Sanger sequencing is really only like one cycle, correct? Like we want to take our DNA, denature it, But the templates anneal and then extend. So one cycle, PCR is usually about 30 cycles, we're repeating over and over again, because we're trying to do amplification step. And Sanger sequencing, of course, we have these DD NTPs. PCR, we only have DD NTPs. And these are the main differences between the two. But the results are extremely different. One of them gives us amplification based off of a primer that targets a specific sequence. The other is able to give us a sequence because we end up running out each reaction on a gel. So that's another way you can do the difference. This is four reactions. This is one reaction. Okay, so you turn in mind how the two differ. Okay, yes.
If for Sanger sequencing, since we're trying to figure out like what the sequence is, how are we selecting the kind?
Okay, so say this is a vector that I cloned, you usually use a primer that identifies part of the vector. So that's one way. The other way is that you design a whole bunch of random primers and then hopefully they all anneal somewhere to G. That's kind of how we use next generation sequencing. So don't worry too much about how we design the particular primer. Depending on what we're looking for, there's multiple ways to actually get this. It's usually either vector based or random primer design. Yes.
That's a list, but you put it into the
Okay, let's make a list of the reaction materials. Okay, so what's the most important thing? What are we starting off with? Why are we using the Sanger sequencing? Yeah, we need primers. The primer anneals to templates. What else do you be needed?
NTPs.
DD NTPs. What else? The NTPs. And then what else? Anything else you need for PCR? You need polymerase. Okay, so we actually talked about this in class. Like, do we need ligase for Sanger sequencing?
No, because it's automatic, it just can't.
Exactly. So there's nothing we actually need to ligate together. So you don't need ligase. So basically think of the things that you need for PCR, but you got to add in DD and TPs. And you know what you need. Okay, question here.
So, when it's adding the A's, the C's, the G's, the T's, is it only adding DDNTPs or randomly adds it and stops it?
It randomly adds it, and that's what stops it. For the most part, you're adding the D and TPs, the regular ones. And every now and then you get something that makes it stop. Yes.
Sorry, could you briefly go over the, like, how you determine which the top or bottom, like the 3 times? I think I vaguely understand it, but I can't tell if I'm like.
Well, we don't want to vaguely
convincing
understand.
myself that I understand.
You need to be
Like,
convinced.
understand the
I'm
exam.
confident. Okay. So, you know, I'm not even gonna write the. Yeah, let's do A. It doesn't matter order, A, T, C, G, okay? So say we have all of these.
Yeah.
And then say there's some bands. And there's bands. Okay, so there's bands on this gel. And I asked you which side of the gel represents the 3' and which one represents the 5'. So if we come back to this example here, we are synthesizing from 5' to 3', 5' to 3', 5' to 3'. So if I'm synthesizing from 5' to 3', and I have a short, small product, where does that short, small product end up? At the bottom. This is at the bottom somewhere. And then so as it gets longer, where's this ending up?
Oh, towards the top, which is, oh, I see.
Mhm.
That's a lot more straightforward than I.
There we go, so
Yeah.
top is 3', bottom is 5'.
Yeah, thank you.
And you can forget, just like mark in your notes, make your notes accessible. Okay, so now that we know this is 3' and this is 5', right? We can essentially just mark the bands in order, and whatever order they appear in is the sequence.
Mhm.
And then for the parent strand, the one we're trying to sequence, the template strand, just write the reverse complement. And when you're taking your exam, take the time, like literally just draw it out, like don't do it in your head. Like don't do typos, don't do that to me. Like I'll give you a point back for a typo, but like don't do that to me. Just take your time, draw it out, make sure it's accurate, check it, and then put it in. Yes.
So reverse complement basically be really similar to the template strand.
So that's what we're trying to get. We're trying to get what the template strand is. So say, for example, I give you a gel. What's the sequence of the template strand?
Perfect.
The reverse complement of whatever this is. Okay, clear on that?
I just to be clear, you said that like when there's a concentration of you said that it would still, that can still like running right? It's just it has a high quality as a high.
So think of this as like an optimal concentration, concentration that has been optimized. And then say I were to ask you, what happens if I put too much VD MTPs?
It would just find
Yeah, we get a whole bunch of these little short mofos. And this, and then we wouldn't be able to get the sequence. That's what would happen. Say for example, I put, or I just forgot to add VD NTPs. Would I be able to do any sequencing?
No, speed really low.
Yep, exactly. We would just get everything all up here on top of the gel.
Mhm.
I'm like, oh, that's so useful. I know, like, maybe the last for nucleotides. Yes, question.
So if it's saying sequencing, with the longer chains, so this is the first reaction, say for A, goes on the T. Does the T fill in the gaps that A has?
Yes.
Or is its own separate, like primary?
It's its own same primer, but separate reaction. But because the T's are in different position, it fills in the gaps.
Okay.
Because it's basically the size, the nucleotides go in order. So the gel is going to be in order. And so once you go through all four, every single gap should be filled in.
Like the chain.
Yeah, and the chain.
How long it is.
Exactly. Okay, anything else on safer sequencing? Thank you.
So for shows up here, is like value. So we don't need it when we're trying to like get DNA from bacteria and then put it into yeast with the process. It's like putting DNA into different species. We need it.
Okay, so shuttle vector is basically for final expression in a host that isn't bacteria. The reason being is because we need to amplify the DNA, and bacteria is the best model organism to use for just amplifying the DNA. So this is why we need a shuttle vector. So if we're designing something and we want to eventually express it in human cells, we need a shuttle vector. Want to express it in yeast, we need a shuttle vector. Because our intermediate is always this bacteria cell that is going to be used to amplify the DNA.
So there's like no shuttles, but let's say there's a question saying that we want to see like it, this thing that could open like when it comes to bacteria. If like it specifies that there's like no shuttle vector being used, we can assume that they have transported it to the human cell.
Yeah, correct, exactly. So even if it's a bacterial product, we have to add in the components that's needed to express it in the human cell. You're still expressing organ replication, selection markers that work in both species or organisms. Yeah, only time you don't do that if you're only working with bacteria. Okay. Cool. Okay.
When we're talking about fermentation, how many big times?
Anybody feel like they can answer this one? How do we define a point mutation?
I guess it's just like a mutation at a single point, like in a nucleotide or base, in a sense, or it can be base or nucleotide that's changed or removed or on one, just one.
Yes, so one nucleotide different from wild type, that's what we mean by point mutation. And multiple types of, I guess, mutations can cause a point mutation. So say we had like a deamination that results in a type of point mutation. So it's just when there's one nucleotide that is changed as opposed to many nucleotides.
But based on that, so it was like, so like, 5, and it's like, this sentence, and shifts, nonsense, transmission, that can say that. It's also longer.
So a point mutation is a change in the nucleotide. The nucleotide has to still be there. A frame shift occurs when you lose nucleotides, to kind of differentiate that. Okay, is that clear for everyone? Exactly. So deletion, insertion, indels are their own category. Point mutation that still has to physically be there and change something.
So it's not a removal.
Yes. Okay, that's a good thing to clarify. I'm glad you brought that up.
Can you explain like the whole how this is like using to knock out, like, when you're your lecture.
Okay, so what you gotta know about homologous recombination and gene knockout. So we're gonna say this is the gene of interest in the genome. Doesn't matter what it is, gene X. It is surrounded by some DNA. So what matters for knocking out a gene is using a vector that say has whatever your marker is, it does not matter. And the idea being that the DNA that's surrounding this marker matches the DNA surrounding the gene. As long as they match, you can get homologous recombination, meaning that they're gonna swap places. That's literally all you gotta know. So homologous genes around, or homologous sequences around the gene and the marker will allow them to swap the homologous recombination. That 1 point clear. Okay, good. Okay, so everyone's got their 1 time, basically we'll free for all until you run out of questions. There we go.
So, but this is on like the reading, I have got a few quick ones. On GFP would say it's a screenable marker or selectable marker? It's
It's
screenable.
readable.
And then, for the question I asked, like, are all the genomes of our cells the same? The answer is yes. But if we were to make a cDNA copy from our cells, all the cells produce different cDNAs because different genes are expressed differently throughout the body, correct?
Yeah.
And cDNA is chromosomal DNA, correct? Or cDNA?
No, so
Oh.
the cDNA is after there's like RNA expression and then basically reverse transcribe it.
Okay,
And for the exam, you don't need to know this concept. But that's what cDNA is, and you'll see that like in a lot of laboratories where different cells express different genes or turn on them, actually express them, and then when you make the cDNAs are different by cell type.
got it. So I guess that's another big reason is like why we want to convert mRNA to cDNA instead of just, sequencing mRNA that we get library, just because cDNA, I guess, is more stable because it's DNA and it can be amplified using PCR.
Exactly.
And then when it comes to Peter Lebon's method, do I know about Peter Lebon? And then how fluorescence works, is that just kind of like that idea where like cell absorbs light at one leg and emits the other, and then you can see that under UV?
Exactly. Fluorescence, you can stimulate it one wavelength, emits another wavelength, and we see it, and it's beautiful.
That's a question on the quizzes, but like.
Okay. Any other like major concepts? Yes.
I just have like a clarification, so if there's an error in the DNA polymerase which is needs to repair, if you have deamination or oxidation, we use phase explosion repair. And then if we have like UV damage or like TT donors, we would use API.
Yes, 100%.
Okay.
Okay, so we're gonna rewrite this up here, so everyone was very, very clear. So an error for DNA polymerase, we end up using.
Oh, yes.
You like go back to your notes. Error in DNA polymerase, basically using mismatch repair. If we have like deamination, like hydrolysis, something that impacts the actual base itself and changes it, we're gonna do base excision repair. If we have, what's the third one?
Oh, UV, TT
Like
donors.
UZ causing binding dimers, something structural, we'll have nucleotide excision repair.
And then we're oxidation.
Yep, oxidation, you have this here.
My phone.
Okay, yeah, I need everyone to have it clear.
Those are base mutations, DMH, not oxidation.
Yes. This one should be the most straightforward. The base gets mutated, do base decision repair. Yes,
Deamination and oxidation, I know in class you remember how silicene gets the end to the so remember over like 16, then we also went over one of the oxidation examples, there was a bunch for other nucleotide bases, we need to know those in the signal of that.
no, you don't need to know that. We went over the two that are most common and the pathways that actually result in a transition or a transversion mutation. Know the difference between those words. What results in transition mutation and what results in transversion mutation.
So deamination is deamination and oxidation.
Mhm. Okay, deamination results in a transition mutation. Once you have like replication, if you don't fix the error in time, oxidation can result in a transversion mutation if you don't fix the error in time. Yes.
What are you getting?
Okay, so let's draw out. What happens in these? So we're gonna talk about deamination. And oxidation. Yes. Okay, so if we deaminate a cytosine, it can become a uracil. If we don't fix this error in time, what happens is that the next step down is we start doing replication. So we have a CUC over here, we have a G. This side will get paired correctly. That one's fine. This one will get paired. A G, the U will match with an A, and a G will get paired. And then when it goes back either to replicate or to get rid of this uracil, what is going to put in its place? A thymine. To end up having a CTCGAG. So when your C to G becomes a T to A, that is your transition mutation. That's clear how that happens. Everybody got a copy, so I'm about to erase them. Okay, transition. Now we're gonna look at another case that is highly similar. A-oxoguanine, if we do not fix this, and then we go to do replication, now we have a G, so G, and a C down here. This one has no problem, you put your G's back. Over here, this isn't a problem, but then you have a 50-50 chance of it pairing with either the correct base or with A. So if we end up getting an A, then what happens in the next step?
I guess G becomes T.
G becomes T. So now, I'll go ahead and erase it. G becomes T once we fix it because we don't want an A-oxoguanine because that's not normally part of our basis. So now you have a G to C that became a T to A. So basically, it's the flip. You're still transversion. You're still basically getting the same two base pairs, but instead of it being CTGA, there's an inverse. Yes.
So if it just transitions to genetically and transitions to
Exactly.
What level this should we know for the examples, the level that deamination usually is transition, oxidation, transversion, disabled DSL, or also the mechanism. I mean, for these examples, we went over in class, we should know.
Yeah. For these examples, you should know like how this mutation happens. And if you know how the mutation happens, you can answer any question about it.
What about the webinar and plus, you know, if I didn't get it.
No, but it's probably still there. There's a class after us.
Okay, I'm gonna-
Yeah, those are expensive.
Thank you for your
There's a-
time.
Question on the quiz, or quiz A, but this was, it was a question about restriction enzymes. So I understand like these typically cleave palindromic sequences and,
Yes.
and I guess evading viral DNA as it's in the textbook. But, how come they don't always produce sticky ends? Or I guess not all restriction enzymes produce sticky ends?
Okay, so there's a table in your reading that basically demonstrates that every single restriction enzyme recognizes a particular sequence and has a particular function.
Okay.
It's all specific to the restriction.
And then I guess they're not necessarily for joining two DNA fragments together. I guess there's different ways you can join two DNA fragments together. Restriction enzymes are not only the most necessary.
So restriction enzymes, nothing to do with joining them together.
Oh,
Everything to do with cutting them up.
okay.
Ligase joins them together.
Ah, and then are found in a wide variety of mammalian cell types? No, yes.
No. Where do restriction enzymes come from? Remember from reading at all? Yeah, yeah, so I can borrow the best mechanism.
Thanks.
Okay,
Thanks so much.
yeah,
Your question from the faculty here, so it was about how you want more volume of the info, so like if one is on an operation, then you use a lot in PCR.
okay, so we need more volume of chromosomal aberration. Oh, no. So first thing, how large are chromosomes? How much genetic material is this? Yeah, a lot. A lot. What is our choice for amplifying DNA if there's a lot of DNA to amplify?
Like putting it in the activity.
Yes, putting the bacteria. It's a way better way of increasing the amount of genetic information you have. PCR will freaking take forever. On top of that, it'll be inefficient because after a period of time, like it's just it's very long. So remember, like in vivo, in the cell, you have this beta clamp that increases processivity, so you can just keep on going down the strand, like go. And in PCR, you don't have that.
Was it because chromosomes are bigger, that's why?
Yeah, because they're big. It's just a crap ton. So you have to know the chromosome is big. And you have to know that when it's a lot of DNA, you don't want to use PCR. Any other questions? Yes. Yes.
So, okay, segment, if the question just says like segment of DNA, would PCR be the better option?
And like, if I would phrase it that way, I'd say a small segment. Yeah, my goal is always to make it unambiguous. For some reason, it's ambiguous, sometimes I fail, then I usually throw out the question. Yes.
For the question we're asked, like, which of the following best describe the first experiment to join two pieces of DNA in the lab? I mean, do we really need to know the story specifically, like how they use like linker DNA of A and T of poly A and poly T, or just that, or just no blunt ends, sticky ends?
Just no blends to hands. The whole story is really cool, how they used to have to add like the poly A's and poly T's to make this happen. And I think it's a beautiful story and worth reading, but you'll notice on the reading recap, if you go watch it, I might just ignore it and keep going on.
Oh, same with the GFP story.
Yeah. Like, I think these stories give really good context of helping you understand, but don't spend your time studying the stories. Yes.
So, blood ones, you said it was like, specific in the clinic, whether it's the penis, could you compare it to the?
Yeah, remember blunt ends, I need two pieces that also have blunt ends to stick together. And this whole idea of blunt ends coming together, this is what happens in the non-homologous end joint. It makes two flat ends so you can just stick anything together. So same concept. Yeah.
I'll go back to that idea with when you do gel electrophoresis and you visualize your results for Sanger sequencing. If you have I guess an equal amount of DDNTPs and DNTPs, you get, I guess you only get like short fragments. I guess maybe it's because like there's too much DNTPs.
Mhm.
And then what if you have like so much DDNTPs? Like you'll practically get like
Just only tiny fragments, so probably hit the first time and
even, and then also you still get tiny fragments relatively like when they're equal. So the idea is
okay, yeah.
increasing DDNTP by to equal or even more to DNTP is bad. You're only gonna get short.
You only get short fragments, not be able to sequence the whole thing.
And the reason for that is kind of what you showed with like that first.
Yeah, so it's just all stochastic. I like that word, it makes me feel smart. Okay. Other questions? Okay, so equal amounts is basically, think about directionality, right? You know you have to have little, you know you have to have less. and that less is important. So when you increase the concentration of DDNTPs, you're always going to inhibit the ability for there to be extension, and you won't get full sequencing. That's always gonna happen if you go in that direction. Yes.
Yeah,
I have a question about the question. I understand it kind of based off like how the average definition. Like there was most of the wording of the question. It was a question where it's like, we'd like to study about the difference between the scheme, the team. And then when the team makes their cells so clean, do you want to make an instruction vector that will allow you to transfer themselves? And then something two, the design of vectors to also have continuity resistance. How can you confirm that your plasmid is properly constructed? I understand all the rest of the defensive choices. The one that I usually cared about was transforming into bacterial cells. I understand that like transforming into bacterial cells is just expected that the vector is transformable. But the question specifically asked about human cells. So I was wondering why bacterial cells wasn't the correct answer choice.
Okay, so question is that, I want you to think very carefully about the actual thing that's being asked. So read out to me the question part, the question.
How can you confirm that the plasmid is properly constructed?
So confirm that the plasmid is properly constructed. We want to make sure that everything that we made is what we think. The things that we added in were pinching, ampicillin, and whatever is needed for expression in human, whatever is needed for expression in bacteria. How does just putting it in bacteria confirm these?
Bacteria and amplify in bacteria and also in sequences. I think the last part of the message is sequences.
So what else does it say in the one answer that's wrong? What does it say in that answer?
What answer that was wrong was incomplete on our answer guide. This was transform the vector into new cells and select this media containing eggs.
Man, nobody told me that was incomplete. Okay, if you ever run anything incomplete, this is like, this merits an email, just tell me, it means it was type or got deleted or something. Okay, that's the problem, but the one you have a question on, so you said, why isn't just transforming into bacteria a correct answer? That's what you're asking.
Transform the vectors bacterial cells, allow the cells to grow, then isolate the DNA and sequence.
Yes,
that's correct.
that's correct.
Yeah.
But A is incorrect.
I understand.
Oh, sorry.
I just didn't like it.
Oh, I was like, but why is A incorrect in T? Well, you used to tell you A.
No, you just know second.
Okay, I'm confused.
I was confused on why bacteria was correct because of the part where it says like transform the result. And then from my interpretation of the question, I thought like proper construction of plasmid meant that. So I kind of just.
Okay, so remember for proper construction, we actually did an example like this in class. Like sequencing is one way, checking, like doing the whole selection process is one way. You do selection process in bacteria. You do the selection process in the human cell, that's the target. You could also do like PCR to like amplify certain sections of your plasmid. So you're confirming the actual genetic construction or manipulation of this plasmid. Even though the target is expressing it in human, doesn't necessarily mean that you only have to put it in human to check the plasmid.
But if the question was worded differently, if the question was worded to say like, how can we confirm that this exact like it was can be successfully transformed into human cells, the bacterial interface wouldn't be correct.
Yeah, correct, because you want to actively check this going to work in human cells. Yes,
Another one quiz was, which of the following is true of the Sanger method? So, it says that, so it does require a primer for polymerase to extend and incorporate, it does incorporate such DNTPs using DNA polymerase, which I understand. It doesn't require, okay, yeah, no RNA template is required, it uses a DNA template, correct?
mhm,
And then, the fact of the matter is that DDNTPs are lacking a 2' OH and a 3' OH,
Yes.
correct? And, last one. The sequencing reaction products are separated by affinity chromatography. Is that not what's going, wait, what is that?
so that's the one when they do the capillary. So this is the more modern method when they mix the 4 different ingredients together. So
Okay.
we won't have the affinity chromatography on actual.
Okay, so,
So it was in your reading.
Yeah.
On the slides.
Yeah.
For the exam, focus on the old school Sanger sequence.
okay, so don't worry about it, okay.
Yes.
I guess this question is a lot. It's like the telomerase in the control.
Okay, so we're good. We have about 7 minutes till 50. And then so I'm gonna go through the telomerase question. That is probably our last thing. And remember, there's gonna be exam review tomorrow. Okay, my brain stopped working. I did not do today. It's long story, broke down cards, good stuff.
Oh,
Yeah, we're struggling. So, telomerase. The whole entire idea behind what the gel looks like is based off the fact that we want to know what telomerase as an enzyme does. So we set up what we call an in vitro assay to test telomerase activity. For an in vitro assay, we're starting off with some DNA that has a 3' overhang. And then so we call this in this type of experiment primer DNA. It kind of sounds weird like a misnomer, but it's primer DNA for the experiment. We also add in proteins that we isolated from cells. In addition to the proteins that we isolated from cells, we want to make sure that we have DNTPs that allow for extension. So you think about the way telomerase is supposed to work. If this group of proteins happens to have telomerase in it, then telomerase will bind to this primer. It has its own RNA primer here, and it will extend from 5' to 3' the DNA that is there and repeats depending on whatever that primer sequence is. So telomere sequence differs by organ, but that's the whole purpose, that this telomerase is going to increase or extend the DNA. So then when we go to run it out on a gel, if on this gel, telomerase actually worked, what we'll see is these repeating units. So we're going to start at the bottom of the gel, small stuff. The primer, of course, remember, says will be small. Telomerase work, we're adding on repeats of DNA towards the end, making it longer and longer and longer. So that's we're getting extension there. That's how the gel looks. Why you see this smear, but kind of like with gaps. So it's that clear. Okay. And then so once you understand the setup, you want to know what are positive and negative controls. So telomerase is not expressed in every single cell in the body. Telomerase, for the most part, is going to be expressed in germ cells, like oocytes and sperm, in stem cells, things that will have to differentiate and basically maintain genetic material. We also see expression in cancer cells. So these cells that we know express telomerase make really good positive controls. But terminally differentiated cells, like skin cells and most blood cells, like a lot of different types of cells, terminally differentiated, they do not express telomerase. So for an example, you had a discussion, you have skin cells. And if they don't express telomerase, this makes a really good negative control. So you can see how we're looking for things that won't have telomerase, things that will. And then in their experiment, what they decide to do is try to figure out if they can take skin cells and turn them into stem cells. And the samples that they did this to are their unknowns. And by having, say, a negative control, so let's say that's skin cells, and let's say this is stem cells, and then your unknowns, it will determine whether or not they're successful based off of the telomerase activity. If they see telomerase activity, they're like, okay, I think their skin cells went towards stem cells. If they don't see activity, it looks more like the negative control, they'll say, okay, you weren't able to differentiate these cells. The other thing I want to point out in this experiment is to also understand what we call additional technical controls. So you'll see in your discussion worksheet that there's 2 lanes that are duplicates. And then there's a 3rd lane where there's no signal. And what they did in order to get no signal is basically, I don't remember if on the discussion worksheet I put if they treated with an RNase or they treated with like a proteinase. But essentially, if you treat it with an RNase before you do the reaction, you lose this primer template, telomerase won't work. So you should get extension. If you treat with like a proteinase for doing this, you basically destroy the protein, you wouldn't get a lane. You could also say, heat up this whole entire reaction mixture so that you denature the protein, you don't get telomerase activity. So any approach to get rid of telomerase activity helps us to know that this activity is specifically due to telomerase. Okay, so is this experiment clear, the discussion? Okay, good. Any other questions before we get off to tomorrow? Yes.
I think DNA, DNA nucleus, we have DNA. If there are no temporary.
If you put the, if you put DNA nuclease, so what happens?
So all the,
Oh, it's everything in the life?
Yeah, so you added the DNA template, you have all the protein lysate, and you added the DNT piece. If you added in a DNA nuclease, you cut up your template. If you added the ribonuclease, cut up your RNA primer.
Also, we're referring to the life that you're referring to the bottom of the top.
Yeah, referring to lysate, I'm referring to the proteins from the cells. So made a lysate of the cells and then isolate just the proteins. We're only adding the protein from the cell. There's no genetic material from the cell. Okay, is that clear? Okay, good. Okay, so, I thought it's going your fingers.
I didn't realize.
Yeah,
I was like, the cap or something.
you know, I like the whole time marker. I'm trying to take the cap out. Okay, let's stop this.
Thank you.